We have levels too, right?
Yeah. Yes, that's what kinetics is. So, and what they do is chromosomes shorten with each replication, and the telomeres are sequences that are there to be lost to protect the genes. And it isn't that actually the chromosomes shorten and shorten and shorten until they chew off a gene and then things stop. It's that there's actually a predetermined length that cells will actually go into senescence. They will stop dividing at some length of telomere. And they don't really know what that is. We do lose about 100 to 200 base pairs with each replication. So why do telomeres, or why do chromosomes shorten? Okay, and what I want to do here is let's think about what this looks like, or the process of replication. So let's say this is a really short chromosome. All right, because we're going to have be able to see both telomeres. You know, in reality, these things are incredibly long compared to how wide they are. But what you're going to do for a round of replication is going to be like PCR. And you should be comfortable drawing these sorts of things, because you should be able to do it think about it for PCR, right? And replication, similar process, not identical, right? We saw the proteins involved in the last one. But let's say we denature. Okay. And our denaturation is really, it's a replication fork, right, that goes along. But if we denature, we'll get two separate strands. Okay, now we're going to add our RNA primers. Remember, they're RNA. And so those primers will go on the ends. So imagine, we'll end up with primers added all along, but on the very end here of each of these chromosomes, you will have a primer.
Question.
Yes. What's the question? There's no bless you is what it was. I got it. All right, so we add those primers, right? Now we're going to extend primers. So far I'm just drawing what I've got above, and then I'm about to extend them. Okay. By the way, I'm doing freeze thaw after having done induction and things like that, so I have wild type enzyme for you. So if you were cursing my name while you were waiting for those things, know that I suffer like you suffer. So now we're going to extend those primers. We're going to go all the way to the end here, and all the way to the end. Blue is DNA, green is RNA, right? Okay. And then we have that RNase activity I talked about, right? All right, for what I'm drawing here, I have ignored, Okazaki fragments and doing multiple primers on it, because we just care about what's going on at the ends, and I'm trying to demonstrate that. And so for this top strand, okay, it goes, but then this bit here we don't get. This bottom strand, there was an RNA primer there. Put a 3' there. And then on the next one, no, can't have that. DNA is anti-parallel. And so what we have here is we have lost sequence.
So then you lose the other side of this.
So we don't lose the rest of this until the next round of replication. But if we lose some on one strand, eventually as we repeat these, we're going to lose some when we make copies. Does that make sense? So the next time we use this as a template, I'm not going to draw it out, but if we use this now as a template to make one of these in the next replication, it can only go to there because there's no more template beyond it. So we're losing a little bit each time because of those RNA primers. Because those RNA primers get removed, they're not part of your genomic DNA. Now what we're losing is we're losing the telomeres. Okay, so these are telomere sequences. And that telomere sequence for humans, remember, is, what is it, TTA? Yeah, it's TTA GGG. Okay, so it's repeats of TTA GGG. Okay, so that's what the point is of the telomeres. Now, questions on that mechanism? All right, so you should be able to draw that out, explain why we lose those. You should be able to think about these sorts of things. And it is, like I said, it's great practice for thinking about PCR. I'm not going to make you do it, but this is the sort of stuff I draw out if I'm trying to explain why you get linear amplification for your site-directed immunogenesis reactions as opposed to geometric amplification like you would do in normal PCR. You do these sorts of drawings and then you can see what's going on. That makes sense?
What specifically would you call that? If you had a label for that picture, what would you?
For what was this picture? This would be the mechanism of telomere loss. Let's go up to the top and see what I wrote. Why do they shorten? I guess I can just, let's go ahead and do a mechanism of telomere loss.
Doesn't it seem more like conducive to evolution to just not do that instead of having a separate thing to avoid that? Or is it a way of preventing cells from from dividing too much?
I mean, the answer may be both. So did everybody hear the question or the observation? So it may be both, that this has become a mechanism. It's also to avoid it. I mean, how do you avoid it? I mean, I guess the way to avoid it is just you have your telomerases active, right?
No,
So there's a simple way. You just need to turn a gene on and keep a gene turned on here. So this is I would say this is at least evolved to become a mechanism for timing how many cell cycles you get. And like I said last time, as an individual organism, you don't like the concept that you die, but as a species, it's a good thing you will. It's good for the species, or at least it used to be. I would say it probably still is. Okay, so how do telomeres get built? They get built by a telomerase. Okay, and what telomerases are they are RNA-dependent DNA polymerases. Let me drag this out so everybody can see it. Otherwise known as, what other name do we have for those? Reverse transcriptases. Okay. So what do we mean by that? You should be comfortable with interchanging these two names. You've heard of reverse transcriptases before, probably, but that's what they are. They are RNA-dependent DNA polymerases. In other words, what they do is they synthesize a DNA sequence from an RNA template. Okay? And so what you can do is you can see this right here. So they have a little piece of RNA that's a template. Okay, so we have an RNA template that's held by the polymerase. And what it does is that it comes in and it grabs the telomere end, it lines it up, it adds another telomere sequence, and then it will shift back and then add again. That makes sense. So it's essentially clamping onto the end of the DNA, filling in this 6 base pair sequence, or 6 nucleotide sequence, and then it shift back and then do another 6, and shift back and then do another 6. That makes sense? Okay. And this is it's not the same enzyme, but reverse transcriptases, we see those in viruses as well. A virus that has an RNA genome that needs to make a DNA sequence to insert into your genome, let's say. Questions on telomerases? Okay. And like I said before, these are active in embryonic cells and cancer cells. Okay, so one of the mutations that cancers need to get going is they need to activate their telomerases if they're going to continue to divide and divide and divide and divide. Otherwise, it's only about 50 divisions. So this is having these telomerases turned off is one of our gates against cancer. Questions? Okay, so there's an interesting, when you get these repeated sequences with all these G's in a row, you can form some interesting structures. These are called G quadruplexes. We're covering this as much because I was trained as a DNA scientist and a DNA chemist originally, and they're pretty cool. So telomerase sequences, or telomere sequences, I should say. And these are single-stranded telomere sequences, right? We've got a single-stranded end hanging out there after that. Go back up to our mechanism. We're left with a single-stranded end, and it doesn't get chewed down or anything to make a double-stranded end. So let's say single-stranded telomere sequences. That's a G quadruplex. And this is what happens. You get 4 G's that are hydrogen bonding with each other. So let me take this picture and highlight. These are hydrogen bonds. They are not covalent bonds, even though they're drawn the same as all the other bonds. And if I take that single sequence with the GGG in it, right? It's got GGG and then what was it? TTA. So we go, you can imagine TTA, GGG, okay? TTA, GGG, TTA, GGG, TTA, GGG. And you end up with a metal ion in the middle. It's usually, I think potassium is best, but it goes, so let's go G, 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 and then we go T, T, A, G, 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 T, T, A, G, 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 T, T, A, G, G, G. And so what they've shown is that with these sequences, I want you to look at B here. What you end up with is your chromosome. We've got at the end here, this is all telomere, let's say, all telomere sequence. And on that single-stranded end, then you end up, these are each of these is a G quadruplex. So you end up with G quadruplexes, not necessarily just two. And then there end up being these DNA, RNA, or they end up with RNA G quadruplexes, which is new. I didn't learn that when I was in grad school. People don't really know exactly how these fit in. It's a stabilization of that single-stranded bit, right? It's going to help prevent, the thought is it helps prevent an exonuclease from coming in and degrading that. But people haven't really shown exactly, they haven't fully elucidated what's going on there. If that makes sense. The biological reason for them. They do exist, though. Questions?
What do we need to know?
What they are?
Just that.
What do you need to know? G quadruplex looks like that. If I showed it to you, you'd be able to name what it is or talk about these four G's together. Okay. You don't need to be able to draw it. Okay.
What do we talk about before these two?
And then other purposes to make it stable.
It's going to stabilize that single-stranded sequence is kind of what is thought there. But nobody's found, like, so telomeres, big deal, right? The telomeres in general, big deal, protects the genes from being degraded. Got a real reason why those exist in nature. Why do these exist in nature? I mean, clearly nature has chosen something with 3 Gs in a row for those telomere sequences, because if you look across various organisms, the sequence changes, but it's still 3 Gs. It's still able to form these G quadruplexes, but nobody's found out really what the function of those is yet. Outside of maybe protecting, but nobody's even demonstrated protecting this bit here. This bit. This is these telomere sequences as well, protecting that from exonuclease activity. And when I say exonuclease, what's a nuclease?
Don't exonucleases like cut out sequences?
They're gonna cut DNA. So let's talk about what's a nucleus. So a nuclease is going to hydrolyze the DNA sequence. It's gonna be hydrolysis, right? We use water, but we can just say cuts DNA sequence. Okay, and it's going to be hydrolysis. I'm going to just continue to say cut. Hydrolysis is the reaction used. And we have two types. We have exonuclease. Okay, that's going to cut from the ends. And we have endonucleases that cut in the middle. Or we could say in the middle. Maybe that's a better way to say it. All right, so when we think about, you guys did plasmid mapping in first semester, right? You used a couple different enzymes there. Those were endonucleases. They cut in the middle. You didn't have any ends because you had a circular plasmid. So we haven't really used an exonuclease in lab. The DPN1 that you guys used is still an endonuclease. It's cutting in the middle when there are not methyl groups, right? When there are not methyl groups. Yes. No, when there are methyl groups, sorry. DPN1 cut when there were methyl groups. Okay, questions before I go on? All right, so we're going to now go on to talking about DNA damage. Got pictures here of, I can't tell if it's the same boy or not, but one or two kids that have a disease that's called xeroderma pigmentosum.
Oh, Dr. Joanne.
Yes. Okay. And what this is, well, I'll go ahead and I can describe it. So, yes, we have, it's Dr.
Sweet.
Dr. Sweet has a daughter that has this. Her daughter went to school with my daughter for a while. When she goes outside, she has like a moon suit she's wearing to keep all the sun off of her. And then when she's inside, they have to have the glass coated with certain windows. So if she's going someplace, they have to have someplace that has, the glass has coatings that doesn't let UV light in or anything. What's going on with this is a genetic disorder. And it's autosomal recessive. You see this in 1 in 100,000 individuals. Okay, it shortens lifespan by about 30 years. What we see here in terms of all these freckles, that's all skin cancer, this stuff, yeah.
So they're different from like normal freckles.
They are, or at least some of them are, right? The freckles might be normal freckles here, but you're much more likely to develop that into skin cancer. If you're more freckled, you actually are more likely to get skin cancer, period. And half of these individuals have skin cancer by age 10.
Because they not produce like enough melatonin.
It's actually they're missing repair genes, which is what we're about to start talking about, DNA repair. So, one of 9 genes involved. It's one of 9 genes involved, and we'll talk about what excision repair is in a second. Okay, and I can't remember whether it's 1 of 9 or whether it's a specific 1 of 9 or whether it's just one of those 9. This is an example of, so for Dr. Sweet, I remember reading an article where they were interviewed and, like the doctors missed this, right? They didn't catch what this was right away. But as a physician, I don't really blame the doctors because if you've got a 1 in 100,000 rate, what doctors are trained to do is, I don't know if you guys have heard, it's horses, not zebras. That they're trained to look for the horse, don't look for the zebra in terms of diagnosing stuff. So you see some symptoms, you should go for the common stuff first, and then once you've ruled all that out, then you can work out your way onto zebras. But that's certainly how you want your physicians to react to things. Anyway, it's a tough disease. So we're going to talk about a little bit, we're going to talk about excision repair after I give you another example of where it's used. I'm going to give you a specific example of where it's used here. So one of the types of damage you get in DNA is called, one of the very common types is called deamination. Okay, and this happens, this is not really a light-induced type of DNA damage. So the excision repair, which is also going to work on these problems as well, is some other form of light damage. I'm not going to actually, I'm sorry, I'm not going to actually be able to tell you what that is. I can tell you about a different type of light damage, but it isn't going to be hit by the excision repair system. Oh, hold on. Actually, maybe I can. Yeah, I will. I'm confusing two of my systems. We've got a single excision repair and we've got a bigger excision repair. So this is a common form of DNA damage. It's caused by oxidation. Okay. So we know that in our metabolism, we are making superoxide radicals, right? We have oxidants that are around, natural sorts of things. You could have more oxidants depending on what kind of drugs you have. If you have Fabism, we talked about that, right? So an inability to make enough NADPH in our ribose or pentose phosphate pathway. That is caused by oxidation, and we can see some of the things down below. And what's really interesting about this is how we explain why we have T's in DNA and U's instead of the U's that we have in RNA. Okay, so if we look at this, we can see all these cases. What we've got is we have an amine that is being replaced with an oxygen in all these cases, right? Okay. But if we look at uracil, right, C, this is cytosine, this is our C in DNA, all right? If we took a C and we damage it and we turn it into a U, right? So if you had a C, if U was used in DNA, how would you know whether you were looking at a U, how would you know if it was a natural U versus a damaged C? Okay. Let me actually put some context, a little more context on this up here. Caused by oxidation, and this occurs 10,000 times a day per cell. All right, so your DNA repair enzymes need to be fixing this all the time. All right, so if you're one of those DNA repair enzymes and you're cruising down DNA, if DNA contained U's and you hit a U, how do you know that if DNA was U's and not T's? You hit a U, how do you know whether that U is supposed to be there or not? What was that?
If it's supposed to be connected to one.
You could check across and see what it's connected to, but is what you're looking at, is it a U that's supposed to be there, or is there a G across from it, and was that G supposed to be there? And I'll show you some other things where you, in terms of replication, where that's a problem. But do you see by having a T in DNA that if a C turns into a U, enzymes can tell that's wrong? Did you have a comment there? I didn't quite hear you, but you okay?
Yeah.
Where are you going? Okay.
Like the reason they know it's wrong is because there's not supposed to be uracil in DNA.
There shouldn't be any uracil in DNA. And so if you see a U, that used to be a C and it needs to be made back into a C. Does that make sense? So deamination is thought to be the reason why we have T's instead of U's in DNA. Okay, repair enzymes can recognize, if repair enzymes see a U in DNA, they can recognize that should be a C and correct it. I'm going to do go C, going to recognize. It's going to be a molecular recognition, right? Hydrogen bonds, van der Waals forces, those sorts of things. London forces for you guys. So if you repair enzyme C, a U in DNA, once again, they know, they don't know, they're enzymes, but they know to fix it, to change it back to a C. Does that make sense? Okay. So this is cool. We have an explanation for this thing that you see everywhere, right? We've got T's in DNA instead of U's. Now we have an explanation for it. So how do these get repaired generally? Depends on kind of what system we're in. These get repaired by what's called the base excision system. And I don't know what I did in the study guides, but what I want to do here is I want you to know one of these systems in terms of the repair systems. You need to know what deamination is. Okay, but in terms of, I'm going to show you a number of different repair systems today. I'd like it that you knew one that you could describe, so I'd say describe a repair system in detail. I will probably also on top of that require you to know mismatch repair, okay, specifically. So what happens in this system, though, it's pretty much all laid out here, is that we have in one, we have some sort of damaged base, okay? So let's say this could be, well, could be any damaged base. So this system is more than just for deamination, but let's say it could be, we'll do this as an example, deamination. So this could be a C2AU. Okay, DNA glycosylase, and there are a number of DNA glycosylases, and they recognize different types of damage, comes in and it clips out the damaged base. So it's going to clip out the damaged base, it's going to still leave the phosphate backbone there. We then have an endonuclease. So cuts where? Endonuclease in the middle, right? It comes down and it recognizes that we've got an abasic site. There's no base there. So this is an abasic.
No enzyme names?
I don't really care. But you would need to be able to describe all steps in the process. So an enzyme does this, a different enzyme does this, okay? I mean, DNA polymerase and ligase, I think I'll expect you to know those, all right? But, so we have an abasic site, that's gone. This piece is going to get clipped out here, and then a DNA polymerase is going to come in and fill the rest in. So our DNA polymerase here, we've got a 3' end, right? Okay, you saw before DNA polymerase, it did this to RNA where it came through and it cut out the bases in front of it. DNA polymerases, some DNA polymerases, especially DNA polymerase 1, have this activity. So DNA polymerase is going to start here. That's a 3' end. And that DNA polymerase is going to grab here. It's going to extend out. It's going to chew things off as it goes for a while. DNA polymerase doesn't go for long. It's not very processive. So it goes a bit, it falls off. And then DNA ligase is going to seal the nick. Questions on base excision repair. So this is a way of removing, you actually spend more energy than one base is worth, right? Because you're actually going to, for each of these nucleotides, if it's indeed this many, it may be more. But if it's indeed this many, for each of these, it's 2 ATPs worth, right? Because every time you add a DNA base to a DNA strand, you're clipping off that pyrophosphate. And then that pyrophosphate breaks down to 2 phosphates. But this is designed, so this is a system that fixes one base. So it's to deal with the system, it's to fix one damaged base, not multiple damaged bases. Questions? Yes?
Can you just re-explain that second step?
Where we have the A basic site.
Yeah.
So this AP endonuclease, right now we just need one of these to do this. Glycosylases are going to recognize, each is going to recognize a different damage base. But the AP endonuclease is going to come in and it's going to say, hey, there's no base there. And so it clips the backbone, and now we have a 3' end that DNA polymerase can start from. And then DNA polymerase 1 goes a little ways, and it also chews up the DNA in front of it. And we saw in our video, we saw it chew up RNA in front of it, right? Do you remember for the replication video on Friday?
Mm-hmm.
Other questions? Okay. All right, then let's go ahead. Since we've talked about the importance of T's and U's, I'm going to go over mismatch repair now, and then we will come back to it. So this is mismatch repair. And mismatch repair is going to fix errors in replication. Okay, so when we talk, we're going to talk about replication, errors in fidelity and replication, not today, but we're going to talk about mismatch. We'll come back to fixing this system. So this system catches 99% of mismatch errors. I think it's going to be 99 to 99.9% of mismatch errors. The system we're discussing is a bacterial system, but it relates to things that would happen in eukaryotes as well. All right, so if we think about fidelity of replication, so in other words, how well we make a copy, when this system actually comes in last, but this system is it's going to give us 100-fold or to 1,000-fold better fidelity, right? We just removed 99 to 99.9% of the errors. Does that make sense? So it's a 100-fold increase to 1,000-fold increase in the fidelity of replication there. We can see the whole thing here. I'm going to blow it up so we can look at it in more detail of how this works. But this explains another thing that we've seen in lab. Okay? So methyl groups on DNA indicate what's the parent strand. Okay, so what happens in bacteria is a few minutes after DNA is replicated, methyl groups are added to all the DNA. Okay, and so where this is relevant to what we talked about is, do you remember in lab we added DPN1 to degrade the template DNA? Okay, that template DNA came from bacteria. Say extracted from bacteria. Okay. In fact, it was extracted from bacteria by other students. So every year or most of the time. Sometimes I have to make my own. I can't remember whether I saved people's this year or not. I hope I did. But what I do is people make their DNA, right? They do their site-directed mutagenesis. They send it off for sequencing. If it comes back from sequencing and it's wild type, I take that DNA and I put it in the fridge to use for templates for next year. Okay, because that's wild type DNA. But that's a side note here. What's going on is that DNA gets made in the bacteria, it gets extracted from the bacteria, and they replicated it a while ago, and it's all has methyl groups on it. All right. So in mismatch repair, knowing what the parent strand is important, right? Because if there's an error, you want to know which strand is wrong, right? This is not an error where you end up with a non-natural base. You end up with a natural base, but you've got two things. You've got A paired with G or T paired with A or something like that. Let's go ahead and look at what goes on with the system then. I don't care that you know the specific enzymes here. Outside of, you should know what a DNA polymerase is, and you should know what a ligase is. Okay. All right. So we've got these sequences GATC that occur randomly. What is that? That would be 1 and 4, 1 and 4, 1 and 4, 1 and 4. So you expect to see these every 4, 16, 64. What's 64 times 4? 256. Is that right? 256. So every 256, you'd expect to see one of these if it was random. And so what we've got is every, say, 256 on average, you've got a methyl group on this blue strand, which is the parent strand. On the brand new, just synthesized strand, you don't have methyl groups yet. All right. So now we have a mismatch. Let's say this mismatch is a what is it? It's a G, a C. A C has a tautomer that allows it to get incorporated when it's not supposed to be. We'll see it in something else. But remember tautomerization? We move a double bond in an OH, right, from organic. C can do that and has this rare form that exists briefly. And if a DNA polymerase grabs it, can put it in. So that's one of the main forms. So it's a problem. So let's say we end up with a T, not a T. Yeah, T across from a G. Except it's going to be down here. So you end up with a G here and a T here. Okay, that's a mismatch. So that's what's designated by these little arrows here. That's a mismatch. And so we have these enzymes, as MutS and MutL, can come in and recognize that that's a mismatch and bind to it. Like this. So we get this MutL, MutS complex. So let's go ahead and write that. This complex recognizes the mismatch. All right, we recognize the mismatch. You don't need to know MutL, MutS. But then what's going to happen next, let me get this other stuff out of the way, is these mut-H's are going to be recruited. And you don't need to know mut-H either, okay? But mut-H is recruited. So it binds to the MutL, MutS complex and essentially spools out DNA until it finds a methyl group. Spool's okay? It's not a very scientific term. It's essentially going to suck up the DNA and make a loop, right? We're going to pull it in from both sides. Maybe let's say scans. How about that? Until it finds a mismatch. All right, so we're spooling through, we're scanning the DNA until we find a mismatch. In this case, it found it on this strand first. It could have found it on either direction. It's checking on the 5' end, it's checking on the 3' end. Here it hits on the, if we were reading this left to right, we're going to call it the 3' end. And then mute H is going to cleave the new strand. Okay. So we've talked, have I used the term nick for DNA before?
No.
Okay. So DNA is cleaved on the, it says modified, the one that has, it's the unmodified, so the one that doesn't have the G, or I'm sorry, the methyl group. But DNA is cleaved on the new strand. So it's the new strand. This is the strand that had just been synthesized. And then what's going to happen is DNA polymerase is going to come in and it's going to essentially synthesize up until the nick here. Okay?
And that's it.
And that's it.
Oh.
I'm going to show you another figure here that does the same thing, but shows both strands. And there's a point I want to make about energy here, energy amounts. So we end up, depending on which strand we've had that cleave, actually the DNA is going to come, I didn't have my directions correct. The DNA is going to come this way, right? Here's our 5' end. And we're going to go 5' to 3' in this direction. So if our nick's over here, we come through and we synthesize that way. And this is DNA polymerase 3. It's a highly processive enzyme, which means it can do a whole lot, right? It can cleave a bunch of DNA or it can synthesize a whole bunch of DNA without falling off. Very processive. And then here we had our nick here, and then we moved out to where we had the we're going to get a nick here, and then we'll remove this part and then synthesize it in. And here we have a nick and synthesize it. Does that make sense? So it just depends on where it finds a nick. You're either going from the nick to the mutation or going from the mismatch to the nick. Okay? You don't really need to know this too well. What does this tell you, though, about DNA polymerase 3 that, based on this figure, that makes it different from DNA polymerase 1? Especially what's going on here. What does that tell you that's different from DNA polymerase 1?
It doesn't need a primer.
It what?
It doesn't need a primer.
Okay, neither needs a primer because they've got you've already got DNA down. The DNA is the primer, right? So this would be like I gave you a figure and asked you to explain something about a question. Let's go back and look at what DNA polymerase 1 did up here and what you've also seen it do before. So here, this is DNA polymerase 1. DNA polymerase 1 is involved right here. Okay? It has, actually, let's start here. We go from here to here with DNA polymerase 1. So we have a nick here, and DNA polymerase starts, and we get to something that looks like that. And compare that to this.
More fragmented.
We definitely have a bigger, it's not really shown too well, but it's shown because you have lines, so we have more going on here. We have exonucleases here that need to come in, so the end ends up being the nick, and chew this piece out. Because DNA polymerase 3 doesn't have the activity where it can bite off. If it has nucleotides in front of it, it doesn't hydrolyze them off. DNA polymerase 1 can do that, DNA polymerase 3 can't. Okay. So that's an example. I don't care that you know that about DNA polymerase 1 and DNA polymerase 3, but if I gave you some figures and some information, I'd like you to try to figure something like that out. That obviously would have been a train wreck in a test question based on your answers. That wouldn't have been a good one.
So wait, how do you see that in this?
Because we don't just have here DNA polymerase 3 comes in choose to there, right? We don't go from here to here.
Oh, so
We have all this complicated stuff where we have to chew a piece off. We have this complex. DNA helicase comes in and lines it, while this exonuclease is gonna. And we have two nucleases that would chew it up, right?
Okay.
DNA polymerase one also use ATP?
What was that?
Does DNA polymerase one also
Yeah.
use ATP?
Well, DNA polymerase 1 and DNA polymerase 3 don't use ATP. They use the nucleotides RTPs. Is that what you're asking?
Yeah, I just saw
So-
ATP on that figure.
Yeah. So all of these are going to be, well, here, okay, so this ATP is involved in this process, kind of getting things bound, right? And the fact that we just see kind of, I don't know how many it is here. I'm assuming these things don't need to use ATP to hydrolyze that. It's not usually the case, but maybe. It might be just to recruit them in. I don't know the ATP cost there. But I do know the ATP cost here. So the next, when we fill these things in, okay, this bit or this bit can be up to 1,000 long. So 1,000 nucleotides replaced. And I emphasize this because we talk about how DNA is this perfect copy, right? That you want to pass on. This is for bacteria. You want to pass on your same genes, your same sequence to the next generation, right? We're different from how our parents look because those things get shuffled around, but not because there are random mutations happening all over the place. So you want to keep the fidelity of that DNA the same. You think about us, we're pretty successful as organisms, so we don't want to be throwing random mutations in there. We want to have controlled shuffling of genetic information. And so bacteria, who are maybe not even as specific, being there, they can put out so many offspring, they will replace 1,000 nucleotides. Each nucleotide is costing 2 ATPs worth, right? Because when we think about what we do for replication, that in replication, each one added is 2 ATPs worth. You have a triphosphate, it becomes a monophosphate when it's added in. Is this all making sense, or you want me to go back to a picture? Let's go back to a picture. So when we think about replication here, here we go. Okay, we think about replication. Every time you add an NTP, a TTP, a CTP, or whatever TP, every time you add one of these, you knock off this pyrophosphate. That pyrophosphate gets broken down into 2 phosphates, and to build that up, that cost you 2 ATPs worth of energy, right? To rebuild that monophosphate, if we were to hydrolyze this off, to rebuild what we what we had, will cost us 2 ATPs worth of energy to stick those back on. Yes.
So this is happening like every cell in your body, almost constantly. How much ATP do you use for this? Like, I don't know, how much ATP do you produce?
Right. So this is bacterial, right?
Okay.
But I would expect that you're spending at least this much energy. But it's on this particular process. This is on replication. So you do your replication. You need to make sure you didn't make a mistake. So it's only when a cell divides that it happens. But the bacteria are spending up to 2,000 ATP to do this.
Okay.
If it turns out that it's more than 1,000 away, it randomly chooses one, which is probably not good. But apparently, that's where natural selection is drawing the line. Questions on mismatch repair? So I hope you take away from that. That's a tremendous amount of energy to try and make sure that you didn't make a mistake. So it's really, really important that DNA does not randomly change.
You said this was mismatch repair. Like there's other systems checking it first before it goes to mismatch repair?
That's mismatch repair. There are 2 other levels of kind of trying to make sure you got the right bases in the right spots. And I will go over those, not today.
Okay.
Okay, last one, and then we'll go here. We've got 2 minutes. We're going to talk about lesion repair. And this is an example of This is an example of what is missing for in xeroderma, xeroderma pigmentosum. So this is called excision repair, not base excision repair. Am I spelling excision wrong? Yeah. Thank you. And so what happens, you have some sort of DNA lesion here. This in particular, something that happens often that's caused by light is what's called thymine dimers. So let's do example thymine dimers. And what thymine dimers are is you end up stacked, you have two thymines stacked, and if you get hit with light correctly, you actually end up with them covalently bonding to each other. So I'm not going to go over this specific repair for those down here because we're not going to have time. We'll just skip it. But I do want to show you the picture. Okay, not exactly stacked here, but you can see that we end up with a thymine dimer here. That's what these things look like. And so one of the repair mechanisms for them is this excision repair, and this is the one of the ones that's missing. And so here we'll just follow the human path here. What we end up with is we cut out a big piece of DNA, and then we replace the DNA level, right? So we nick and nick. We have something that recognizes, hey, we've got this problem. We're going to nick across from it. But with DNA helicase, we'll just kind of unwind it out. And unwinds it, and it floats away. And then, in fact, it's a 20 timer, and then you're just going to have DNA polymerase repair, and then seal the nick with DNA ligase. But notice a lot of these things end with DNA polymerase and DNA ligase, right? Okay, I will stop there. You folks have a good day.